Okay, so I made this video just as like a last-minute thing to study for the AP exam. First thing I want to say is take a deep breath, relax. You're not going to do good if you're nervous. You are prepared. You can do this. Okay, so the first thing I want to say is how you're going to take this exam. Part 1 is roughly 63 multiple choice. Last year it was a little bit less than that. And 6 math. Okay, so when they say you may now start your test, I want you to flip to the back and do the 6 math first. One of them's going to be Hardy-Weinberg. One of them's probably going to be rate. It's a good chance one of them's going to be chi-square. We're going to do that later in this video. But do the math that you can. That way, after you finish the math, you'll flip to the front and start with number 1 and do the multiple choice all the way through. Do the best you can on the multiple choice. If you start to run out of time, pick one letter, like for example A, and bubble that letter in for the rest of the ones you didn't get to. Your chances are going to be better if you do one letter all the way down. Okay, 10-minute break. Don't talk about the exam. Don't think about the exam. Take a break. Part 2. The 2 long free response will be first. Those are probably going to be 10 points each. So you want to take your time and do those 2 first. Make an outline. Really think about the question. All year I've told you to answer the question. Focus on those bold words like identify what this is, describe what this is. So focus on that bold word and answer all of the question. Make sure you read all the question several times and answer all of it. Do those in the order that you know best. Then do the 6 short ones in the order that you know best. So that way you're going to eliminate the ones you know, get the points where you can. The goal is not to get all the points, but to get the points you can get. So if you run out of time, you didn't get to answer the one you didn't know. Okay, so the first thing I want to really talk about is for content is the different structures of the macromolecules. So I have 4 macromolecules here. The first one, nucleic acids. And that's anytime you see nucleic acid, we're talking DNA and RNA. It's made up of nucleotides, which is, if you look at this picture, a phosphate group, which is negatively charged, a sugar. If it's DNA, it's going to be deoxyribose. If it's RNA, it's going to be ribose. And a nitrogen base. So remember that DNA has phosphates, like it has a phosphorus, and it has a nitrogen in it. Okay, the nitrogen bases are adenine, thymine, cytosine, guanine, where thymine is a pyrimidine, which means it's smaller than a purine. Adenine is a purine. And cytosine is a thymine. Guanine is a purine. So they pair together. And that's in DNA. In RNA, the T's are replaced with U's, which are also pyrimidines. Okay, proteins are made up of amino acids. Got to know that. Proteins are made up of amino acids. You know that proteins are made in the ribosomes. And they fold, and that determines their sequence. Okay, so here is an amino acid. It's got the amine group and the carboxyl group right here. This R can be one of 20 different things. It can be different things. It's probably on the test, if I had to guess, it's not going to be an R. It's going to be a molecule with carbons and hydrogens, and to make you have to really look at it. So if you see this NH2, it's an amino acid. And if you see this this might be OH on one, and then the O. It's an amino acid. Okay, when amino acids go together, you're going to lose water. So if you take and put 2 amino acids together, or 2 sugars together, you lose water. Okay. And then proteins fold. They fold. The primary sequence is just a straight line of amino acids. And it's got covalent bonds or peptide bonds in between it. The secondary structure is where it coils up with the alpha coils and beta pleats. Think about the pipe cleaner we folded there at the year. The tertiary structure is where the sulfur goes together, the disulfide bridges. So that's the tertiary, and then the quaternary is where more than two primary sequences go together. And that the protein, just remember that the protein is made from the RNA. Like the RNA goes to the ribosome, and because of the sequence of RNA codons, the protein is made. And that sequence, that primary sequence, is really important as to how it will fold and how it will function. Okay, phospholipids. I think I have a picture actually. Here we go. Here's a phospholipid. It's got that phosphate up here. It's got a saturated and an unsaturated fatty acid. The head, remember, is hydrophilic, like a cat likes to drink water, hydrophilic, and the tails are hydrophobic. It's polar, nonpolar. Okay, and then just since we're on this page, the structure of ATP, remember it's got 3 phosphates, 1, 2, 3, negatively charged phosphates, a sugar, a 5-carbon sugar, and then the nitrogen base adenine. So it's just like a nucleotide. It's got phosphates, sugars, nitrogen bases, but there's 3 phosphates, and they're all negatively charged, and this last one is ready to leave and get up. So when it's got 2 phosphates, kind of like the one I have here, it's a DP, diphosphate, but with all of them it's triphosphate. So this is energy. With a TP, it's energy. Okay, so carbohydrates is next. Carbohydrates are going to be quick energy molecules. They're made up of a chain of carbons or a ring of carbons, and they've got H and OH on the ends, alternating H and OH. Okay, so that's a carbohydrate. Quick energy. Oops, that should be OH. OH, H. So that's your carbohydrate. The difference in the carbohydrate structure and the lipid structure. Lipids just have H's coming off of them. Okay, so I'm going to go in and erase all the O's. Now it's a lipid. Lipids are saturated with just hydrogens. Sugars have hydrogens and hydroxides. Lipids are good for stored energy. Carbohydrates are good for quick energy. Okay, and here's another picture of ATP. Just remember it's got the 3 phosphates that are negative, sugar, and then adenine. Okay, dehydration or hydrolysis. Okay, think about the words, and you're going to be able to get this right. Remove water. Dehydration is remove. I don't know why I can't write on this. Okay, remove water. So when you remove water, you build bonds. So look at the picture. You're taking 2 sugars, putting them together, you remove water, and you made a polymer. They're a disaccharide. Okay, so dehydration, you remove water, build bonds. Okay, hydrolysis is opposite. Lysis, break. You add water, break polymers apart. Okay, one of these releases energy, one of these stores energy. Okay, when you break, like when you eat food, when you break the bonds of food, you release energy. Okay, when you build a polymer, you're storing energy, or it's taking energy. So you're putting energy in. Okay? Catabolism versus anabolism. Breaking down is catabolism. Cats break furniture down. Cats. Dehydration would be anabolism. Little ants build up their houses. One of these is cell respiration, one of these is photosynthesis. Breaking, this one's going to be cell rest. This one's going to be photosynthesis. Photosynthesis builds sugar. Cell respiration breaks sugar. So take a second, maybe pause this video and get those words down pat. Lots of vocabulary on this page. Okay, so enzymes are just amino acids. They're proteins that have a specific job. So the purple thing, that's the enzyme made up of amino acids, and it works on a substrate, and the substrate is the red thing. So enzymes act on substrates, and they make products. So there's your products over there. So enzymes are made up of amino acids. They're catalysts. That means they speed up a reaction. Okay, so an example of an enzyme. Okay, so the enzyme that I'm choosing is the purple right here, is CDK, and that's cyclin-dependent kinase. So this enzyme's name is really kinase. Or CDK. Okay, and kinase depends on cyclin in order to do what? In order to start mitosis. So when cyclin binds to kinase, mitosis occurs, or cell cycle occurs. When there's no cyclin right here, then there's no mitosis. So cyclin is degraded throughout the cell division process. Kinase is always there. So when cyclin binds to kinase, it starts cell division. So that's just an example of an enzyme and how it works, and it's also an example of how cell division is regulated, mitosis is regulated. Okay, so let's think, what are some things that would affect the amount of product made? What would affect the amount of the product that's made? So this product is not made as fast because the temperature is different. The salt concentration is different. The pH is different. Or inhibitors are present. Okay, so like if the temperature goes up too high or down too low, the product is not going to be made as fast. If the pH is up too high or down too low, the product is not going to be made as fast. And then inhibitors, those are things that stop a reaction. Okay, so example of an inhibitor, two of them. You have competitive inhibitors bind to the active site, blocking the active site so the substrate can't get there, slows down the product. Okay, the other type of inhibitor binds to the allosteric site, and this one's called non-competitive. This one's called competitive, that first one. When the non-competitive binds, it changes the shape of the active site and still slows down the rate of the product. So it just changes the shape of the active site. Okay, so this picture, I know we looked at it in class, but just want to look at it again. Reactants are put with energy. Energy is put in, and that makes a big product. The amount of energy, look at the amount of energy required right there, stored in the bonds. So this is endergonic, or this is anabolism, or this is dehydration. So just look at this picture and make sure you could identify this if it were to be on the exam. You got reactants and energy going in, making a product. Anabolism, endergonic, dehydration. Okay, this one's opposite. You got a large amount of reactant, so like a large sugar, and you break that sugar down, like cell respiration, and what you've made, you've released ATP. But what's left over is a smaller product. So the amount of energy released. So if you thought about this as like just glycolysis, you take the glucose and you break it down. So you take the glucose right here. You break it down. Having a hard time here. Okay, glucose. Break it down. You release ATP when you break the glucose. And what's left over is the pyruvic acids and ATP. Exergonic, catabolism, hydrolysis reaction. Okay, so this graph, I just want you to be able to read this. So take a second, see if you can figure out what this is saying. First thing you always want to do is look and see what's on the X and Y axis. So you're looking at energy versus progress. Okay, and looking at this one, you got two lines labeled, with an enzyme and without an enzyme. So from looking at this, I want you to be able to see that without an enzyme, it requires more free energy. Enzymes lower the amount of energy required. They lower the activation energy. Okay, the next thing, and I chose chemiosmosis, that word, just because I want you to know, chemiosmosis is how active and passive transport work together to make ATP. Two types of energy coupling, active transport, passive transport, work together to make ATP. Okay, this is a picture of the thylakoid in photosynthesis. You know this picture. Electrons come from water, and they go through the electron transport chain. So the yellow line is electrons. The electron goes through the electron transport chain, binds with NADP to make NADPH. So in the light reaction, NADPH is made. Okay, but the part about ATP, as it goes through, hydrogen molecules are pumped into the thylakoid space. And those are hydrogen protons. So remember that those are positive charged because the electrons went through. So hydrogen protons are pumped through. It builds up a gradient of hydrogens, just like you're blowing up a balloon of hydrogens. Well, the hydrogens have to go somewhere. So they're going to go through ATP synthase. There's the word right there. ATP synthase turns, makes ATP. So the light reaction of photosynthesis makes NADPH and ATP. Okay, so you want to know that chemiosmosis is how active and passive transport work together to make energy. Okay, so here is the active transport. Those hydrogens are pumped in. Okay, it builds up a gradient. So it's going from low concentration to high concentration. Pump. That's active transport. Okay, passive transport is where all these hydrogens go through here. Because think about it, they're going from the high concentration to the low concentration. But it's like letting air out of a balloon. The air goes out this way. ATP synthase turns, makes ATP. Same thing, cell respiration. Only thing is it's upside down. You got electrons coming from NADH and FADH. Right here they are. FADH really lets off right there. Okay, so electrons go through the electron transport chain. So the pink line is electrons this time. You can see the electrons going through. And the electrons bind with oxygen right here and make water. But in the meantime, as those electrons go through, hydrogens are pumped. It's in between the cristae and the mitochondria. So in that outer membrane space, it's pumped hydrogen protons up there. The hydrogen protons build up a gradient. They go down through Big Mama, so they go back into the cristae, turn, make ATP. Okay, cell signaling. The ligand binds to the receptor protein on the cell membrane. Ligand binds to the receptor protein. That's reception. This ligand is just a signal molecule. It's a chemical message. It might be a protein. It might be a hormone. It might be a steroid, something like that. Ligand binds to the receptor protein. And what that does is it makes the receptor protein signal inside. So the receptor protein, when it receives the message, it sends the message inside, but the ligand doesn't go in. The receptor protein just relays it into the cell. The only way the ligand goes in is if it's if the ligand is estrogen or testosterone, a sex hormone. Those go inside, but most ligands, like insulin, doesn't go inside. So the ligand binds to the receptor protein. Okay, and then the message not the ligand. Ligand doesn't go in. But the message is changed. It's just relayed inside the cell. And it's changed into a form that it's translated into a form that the nucleus can understand. And then now the message right here, like the secondary message, is going to go into the nucleus and tell the DNA what the ligand told it to tell. It's going to do the response. So an example of a response might be to turn on transcription. So the DNA should now make RNA, or make a certain protein, or turn on a certain function. Okay, so an example here, let's go with, I'm going to say the signal molecule is IL-2. And IL-2 is the signal that's released from helper T cells to let your other cells know that you're sick. Okay, so helper T cell release IL-2. Now, IL-2 is going to bind to, we're going to say this is a B cell. And it binds right there. And all that message says is, there's an invader. We're sick. You need to start making antibodies, because B cells make antibodies. Okay? So the receptor protein sends that message inside. And it translates that message into something right here. This green thing is what the nucleus can understand. So now the green thing right here, this molecule, goes into the nucleus and tells the DNA to turn on protein synthesis. So the DNA is going to make RNA, and the RNA is going to make a protein. But this particular protein is going to be an antibody. Okay, so that's an example. Neurons. Neurons have dendrites. Dendrites that receive a message. Okay, and then, so that's like the receptor proteins on the dendrites. The cell body is where transduction occurs. The message is changed. And then this is where the nerve impulse goes down the axon. The axon is wrapped in those yellow Schwann cells. It's just like rubber coating on earbud or something. It's a protective coating around a wire, because the axon carries an electrical charge. The end is the synapse. The nerve impulse will jump the synapse and go to the next neuron when calcium comes in the end right here and helps it jump. Okay, so let's look at this graph down here. Okay, so right here, the negative seg of D, that's resting potential. That's when the neuron is at rest. Okay, the going up, that's when the message has been received right here, and it changes. And before the impulse, so the nerve impulse is like right here on the end of the cell body. Before the nerve impulse can go down the axon has to be depolarized. The axon has to be depolarized. I'm going to erase all this and see if I can. Okay, so before the nerve impulse, remember I said the nerve impulse was right there. Before the nerve impulse can go down the axon, it has to be depolarized. And it's depolarized when sodium floods in. So if sodium floods into the axon, because the sodium was on the outside. Remember I told you that the resting potential was like a salty banana. So the salt's on the outside. To depolarize it, the salt's got to come in. Okay? Repolarize, this is when the message is all the way, the nerve impulse is all the way down here. It met threshold, which is negative 55. It got all the way to the top. Action potential occurred. Now we got to get that axon back to normal, back to rest. So we're going to repolarize it by sending potassium out. If it gets too far, if too much potassium comes out, we call it hyperpolarization right there. And then we're back to rest. Okay, so go back to the nerve impulse, which is right here. Okay, so before the nerve impulse can jump from this neuron to the next neuron, calcium is going to flood in and make vessels, or vesicles, like like little bubbles. Calcium is going to make little bubbles for the nerve impulse to be able to jump right here. And the calcium opens up. Calcium's like the ligand. It opens up the protein gates so that the nerve impulse can go inside. Only ligand you need to know for the nervous system, they're called neurotransmitters. So the ligands are called neurotransmitters. One specific one is acetylcholine. That's the nerve impulse that makes you flex muscles. Okay, immune system. First time you get exposed from a pathogen, so whatever you get strep from, so like a bacteria. Okay, so it comes in. The first thing that happens is macrophages engulf it, and then they destroy the pathogen with lysosomes. Okay, you want to know that macrophages, right here, this word, macrophages are innate immunity. They are not specific. You're born with macrophages. They are innate, and they use lysosomes. That was on last year's exam. That was on your mock exam. So make sure you know that macrophages are innate immunity. Okay, then macrophages, they do two things. Macrophages release a message, release a ligand, release a chemical message called IL-1. IL-1. And that signals helper T cells. That signals to helper T cells. And that's the message. It's the interleukin 1, the first message between leukocytes, white blood cells. And it says, hey, we have an invader. Hey, we're sick. You need helper T cell. You need to signal T cells and B cells. So macrophages tell helper T cells. Helper T cells tell B cells and cytotoxic T cells, or killer T cells. Okay, this message right here, the red one now, from helper T to the cytotoxic T cells and the B cells is called IL-2. Okay, the B cells are going to make antibodies to kill the pathogen. The T cells are going to make a chemical called paraffin. That paraffin attacks or drills holes or destroys the infected cells. So T cells kill infected cells. Your cells that are infected. And that we call that cell mediated. T cells, they release almost like bullets. It's a chemical that destroys the cells that are infected. B cells make antibodies. Here they are right here. B cells make antibodies, and the antibodies attach to the pathogen, to the actual pathogen, and they inactivate it. So the T cells kill infected cells. The B cells inactivate the pathogens. So both are really important. Eventually, we'll erase a lot of this. Okay, the B cells, the helper T cells, and the cytotoxic T cells all make memory cells. Right, here's your memory cells right here. Memory cells, memory cells, memory cells. And so the second exposure is much faster. The second time you get the pathogen, those memory cells are stimulated, you make lots of antibodies, and lots of paraffin, and a lot of times, the second time you're exposed, your immune system works so fast, you don't even feel the symptoms. So the second exposure, you make more antibodies much faster, and you don't even see any symptoms. Okay, one other thing we didn't mention is the signal molecules MHC2 and 1. Okay, so these are not signal molecules, but we call them antigen presenting molecules. So it's where the cell has a protein on its outside as an antigen. The cell has a protein on its surface, and it uses that protein to tell other cells what's going on. So it holds up its little hand. Okay, the macrophages right here hold up their antigen, surface protein, is MHC2, major histocompatibility complex 2. Okay, what it is like he holds up a protein like a little flag, holds up a little protein that says, I killed this pathogen. Now, other macrophages go kill this pathogen. So this is to signal the MHC2 is a communication between white blood cells and white blood cells. This is what the pathogen looks like. This is what the pathogen feels like. Go find it. So I've heard before, it's almost like a trophy. This is what I killed. You go kill it. Okay, other cells, like your cells in your throat or other cells in your body that might be infected, they have a little surface protein that they hold up. It's called MHC1. And that one is like, I surrender. It's when I'm infected, kill me, I need to commit suicide, I'm sick. So that's a cell in your body that's infected, holds up an MHC1. A white blood cell holds up an MHC2. So MHC1 is from your body cells to tell white blood cells that they're infected to kill me. Okay, negative feedback, really important. When you see the word negative feedback, I want you to see the word homeostasis. Okay, when you see the word negative feedback, I want you to see the word homeostasis. Negative feedback is how your body regulates homeostasis. Example, look at this picture. When your blood glucose goes up, your pancreas releases insulin. Insulin targets cells, like insulin will go on a muscle cell, and it'll open up the glucose channels, and glucose will go into the muscle cell. When glucose goes into cells, it lowers the blood glucose. And that's going to regulate your blood glucose. Okay, if your blood glucose gets low, like if you eat a boiled egg for breakfast and don't have any sugar, then your pancreas is going to release glucagon. Okay, glucagon, the target for glucagon is the liver. So glucagon goes to the liver, tells the liver to release glucose into the blood. So what's that's going to do? That's if you had low, you release glucagon, it's going to make it back to normal. So homeostasis is how your body regulates blood glucose, temperature. Bicarbonates help you regulate the pH in your blood. ADH helps you regulate the amount of water in your body. ADH, that hormone. Okay. Another example of negative feedback is this one over here to the right. And this is just showing you, the hypothalamus releases a hormone that tells the pituitary gland to release a hormone that tells the thyroid gland. So this is just communication between glands. So the hypothalamus tells the pituitary gland to release the hormone. The pituitary gland tells the thyroid to release the hormone. So that last hormone is thyroxine. Okay, when you get enough thyroxine in your body, thyroxine will actually stop the process from occurring. So this is where the product of a reaction stops a reaction from occurring when you get too much of it. So this is still regulation. Your body, when you have enough thyroxine, it stops making it. When you don't have enough thyroxine, it makes it. This example of negative feedback is specifically called feedback inhibition. So you want to know that word. Okay, this is positive feedback. So all of that was negative feedback. Feedback inhibition is an example of negative feedback. Okay, positive feedback is where a response is amplified. Positive feedback is where a response is amplified. Positive feedback, amplified. Example would be just an example, not the one on the picture, but just one to think about. The bigger a population gets, the bigger a population gets. So like the world. the more like a growing country, the more babies are made, then those babies are going to grow up and have more babies. Then those babies are going to grow up and have more babies. So that's just an example of positive feedback. Eventually, that would be controlled because of lack of food and jobs and space. So that would be eventually controlled. But an example of positive feedback in our body, estrogen right here from the ovaries induces, here we go. Okay, estrogen from the ovaries. Estrogen is a hormone that comes from the ovaries. Induces the uterus to release oxytocin. So when the uterus releases oxytocin, it contracts. And that's what causes contractions when someone's in labor. Okay? The oxytocin, then it's going to, the oxytocin stimulates for more oxytocin to be released. So it's like you're going into labor and you release oxytocin because you're in labor. Well, because you release oxytocin, you contract. And because you contract, you release more oxytocin, and more oxytocin, and more contractions, and more oxytocin, and more contractions, and more oxytocin. So the contractions make you release more oxytocin. Because you release oxytocin, you contract. And because you contract, you release more oxytocin. So it's pretty much like you're in labor, and then you're really in labor, and then you're really, really, really in labor. It's just positive feedback. The oxytocin is amplified because of the oxytocin. Positive feedback. Okay, the difference in protostomes and deuterostomes is, okay, in an organism that, think about it, your mouth and anus are connected. And that's kind of gross, I know. Think about it, though. It's one long tube, and you've got sphincter, so things don't go the wrong way, but it's one long tube. Okay, so an organism that makes its mouth first. So we're all going to look like this, mouth and anus, all connected. This is like really early in development. The organism that makes their mouth first is called a protostome. Okay, so you can see right here it's starting to make the mouth. Okay, an organism that makes their anus first, or I think the textbook says their mouth second, is called a deuterostome. So you can see right here they're starting to make their anus first. Okay, you just want to know that deuterostomes, that's going to be backbone. So fish, amphibians, reptiles, birds, and mammals, we all make our anus first. And echinoderms, that's going to be starfish. So starfish and organisms with a backbone make their anus first. All the other ones, like all those little worms and insects, make their mouth first. Alright, protein synthesis. This is how your body makes proteins. Okay, so you've got DNA right here. DNA, let's go with purple. Okay, so you got DNA right here. DNA unzips, RNA polymerase attaches to DNA, and the RNA polymerase makes RNA. That's called transcription. So step 1 is called transcription. Okay, so step 1 is transcription. That's where DNA makes RNA. The enzyme that does it, shows it in the picture, you have to look real close, is RNA polymerase. Makes RNA. Okay, so now we have this red strand of RNA. It's got uracil, it's less stable, it's one-stranded. The RNA has to be modified. So after transcription is modification. And I think your textbook calls it post-transcription. After transcription, modification. Okay, look at the RNA right here. It's got to have a 5' cap added. So on the 5' end, they add a cap, and it's a 5' methylated cap. And I like to think about that cap as like a hard hat. It's going to help it find its way to the ribosome. So you get a 5' cap, or a 5' methylated cap. Okay, you get a poly A tail. All that means is a bunch of adenines at the end. A, poly A, many A's. Poly A tail on the 3' end. Okay, and then you're going to remove, probably the most important part, remove the introns, remove the junk. So remove introns so that the exons can, see the dark red is the exons? Remove the light red right here, which is the introns. The introns is non-coding. It's the DNA that doesn't make a protein. It's the DNA that's not a gene. So remove the introns so that the exons can go out to the ribosome. So now, right here, we have an RNA that's got a cap, the poly A tail, and only the exons. Only the exons are there. So now that piece of RNA is going to go out to the ribosome. So now you see the red ribbon right here out of the ribosome. Okay, so step 2 of protein synthesis is translation. And that's where RNA is going to actually make the protein. Okay, so the RNA looks like it's going this way. It goes through the ribosome, and the RNA has on it codons. So the 3 letters on RNA is a codon. And as it goes through the ribosome is going to read it like, almost like people can read Braille. The ribosome is going to read the codons. And depending on which codon, it's going to bring a certain tRNA that has an amino acid attached. So the sequence of RNA determines the sequence of amino acids. Okay, so let me say that again. The sequence of RNA, the codons of RNA, determines the sequence of the amino acid. So here's the amino acid you're making, the primary structure of proteins. That amino acid we drew at the beginning. They're all attached now, and they're attached in the order that the RNA was in. Okay, so the RNA goes through the ribosome, and the ribosome builds the amino acid. Okay, after you make the primary sequence of the amino acid, then it has to be modified. So post-translation modification is simply folding. You got to take this thing right here, this amino acid primary sequence, fold it so that it can be useful. It may stay inside the cell and be like helicase, or it may go outside the cell and be like an antibody. Okay, if it was made on a ribosome that is attached to the ER, if the protein was made on a ribosome that was attached to the ER, then it's going to go to the Golgi, be folded, and shipped out of the cell like an antibody. If it was made on a free-floating ribosome, then that protein is going to be folded and used inside the cell and be like helicase or RNA polymerase, or centrioles are made up of proteins. So it's going to be a protein that's used inside the cell. So if it's made on an attached ribosome, it's going out of the cell. Free ribosome staying inside the cell. So transcription, translation, those are the 2 steps to protein synthesis. Okay, DNA replication happens in interphase, the S phase of interphase before mitosis. And DNA replication is how DNA replicates and makes one exact copy. It is semi-conservative. It makes half old. It makes half of a new copy every time. Okay, so if you look, you got topsomerase is the enzyme that's untwisting, but I don't think it made this picture. And then you have helicase right here that's unzipping. I don't guess that's labeled. Okay, so helicase unzips. It takes those hydrogen bonds between the nitrogen bases and breaks them apart. Okay, and then DNA polymerase 3, right here, adds nucleotides in the direction of 5' to 3'. DNA is built from 5' to 3'. DNA is built 5' to 3'. Okay, so DNA helicase unzips it, and DNA runs anti-parallel. So if you look right here, it runs like this. Okay, so when it unzips it, look up at the picture that I'm drawing on the top left. DNA polymerase has to start right here on the 3 side and build it 5 to 3. Okay, and then on the bottom strand, it has to start over here and build it 5 to 3. Okay, so it's starting right here and going this way on the leading strand. The leading strand is built in the same direction that helicase cuts. Okay, on the lagging strand, DNA has to be built this way. So what happens on the lagging strand, watch. It starts right here, and it goes that way. And then it has to back up. And DNA polymerase 3 has to back up and come here. And then back up and come here. And then back up and come here. So it keeps having to backtrack. Ligase comes in and zips those fragments together like this. Those fragments are called Okazaki fragments on the lagging strand. Okazaki fragments are like maybe 10 nucleotides paired together. Okay, so that's replication happens in interphase. And that's how DNA makes DNA. This was how DNA makes proteins. So DNA can make two things. It can make proteins and it can make DNA. Okay, so chromosomes. You know that you have 23 chromosomes in a sex cell. So sperm and egg. That's haploid. Every other chromosome is diploid. So these are just errors in chromosomes. So this would happen in that initial zygote, or maybe in the egg that was fertilized. So the egg or the sperm had errors. And this would be how people have chromosomal errors. Right here, hang on, back up. How people have mutations here is because the RNA or the DNA sequence is different. Chromosomal errors means that their chromosome doesn't have the right amount of genes on it. Okay, so the first one, deletion. That's where a gene is deleted. When your egg was fertilized, you were missing a gene. So this would be how someone is missing a gene. Duplication, you can see BC. BC was duplicated. So you got a gene twice. Or in this case, you got two genes twice. Inversion, that means flipped. When something's inverse, it's just backwards. BCD, over here it's DCB. So it flipped. In translocation, trans is across, location. It moved to a different area. So I always think about translocation like in the crossing over process. When they crossed over, the little piece of gene that was supposed to attach to one of the chromosomes attached somewhere else. So, example right here looks like AB attached to PQR, and MNO, when they crossed over, attached to the wrong place. So think about, how when we talk about the crossing over in meiosis, when they separate, it makes they switch genes. Well, they switched genes, but they attached on the wrong place. Okay, before we start this, think about meiosis. Meiosis is how your body makes sex cells. So meiosis is how you make sex cells or haploid cells. And every sex cell should have 23 chromosomes. Just real quick about meiosis. Meiosis is how we have variation or differences because of sexual reproduction, because of the crossing over, because of the chromosomes separating, the independent assortment, and because of random fertilization. That's how me and my sister have the same mom and dad, and we look different. Meiosis increases variation. Mitosis makes identical cells. Okay, so meiosis, the chromosomes line up in metaphase right here. Here they are, lined up. lined up so nicely. And they should separate and look like this one. But they didn't separate. And see how this one didn't separate? That one right there. So this right here is normal. They should all look like that. But this one doesn't. You got N plus 1, N minus 1, and then N. Or it should be N. This one right here is like N with an extra. This one's N but minus. And they should all be N. They should all be haploid. Okay, so if this is the egg that just happens to drop and get fertilized that month, it could cause a baby, or would cause a baby, to have an extra chromosome. So instead of having 46 chromosomes in every cell, the offspring would have 47 chromosomes in every cell. Non-disjunction, that's how people get Down syndrome. Down syndrome is not something that, just because my sister has a kid with Down syndrome, doesn't increase the chances of me having a kid with Down syndrome. Down syndrome it happens when you drop the egg. If non-disjunction occurred in my body right now, it wouldn't hurt me at all. I wouldn't even know it unless I had an offspring that was affected. So non-disjunction could happen right here in meiosis 1 or in meiosis 2. So non-disjunction is where the chromosomes fail to separate correctly, and it causes the zygote, the offspring, to have an extra chromosome or one less chromosome, like Down syndrome. Okay, operons are how bacteria control transcription, and the textbook normally, or questions normally refer to it as gene expression. DNA makes RNA. Operons are how bacteria control that. Okay, so just remember that RNA polymerase attaches to DNA, and that makes RNA. That's normal. RNA polymerase attaches to DNA and makes RNA. Okay, so look at the top picture. This is your lac operon. Okay, so RNA polymerase would normally attach, like look at the second picture. RNA polymerase attaches and makes the RNA right there. And it does that because this repressor is not bound to the DNA. So the RNA polymerase can go down it. Because the lactose binds to the repressor. So the repressor doesn't bind to DNA. If you look at the top picture, lactose is not present. No lactose. So the repressor right here is bound to the DNA, so there's no RNA. Okay, so just take a second. This is hard, so take a second and look at it. The repressor, think about it real simple first. When the repressor binds to DNA, then DNA does not make RNA, no RNA. And that's what's right here. The repressor is bound to DNA. Okay, so if this repressor right here would come off of the DNA, then the DNA would make RNA. So how do we get the repressor to get off the DNA? Well, it shows it right here. When the lactose binds to the repressor, then RNA is made because the repressor is not on the DNA. So we call the lactose in this picture an inducer because the lactose binds to the repressor and then the DNA makes RNA. So this shows here's the RNA being made, and the RNA makes different enzymes. So DNA makes RNA, RNA makes proteins. This is in bacteria. Okay, so the repressor stops transcription. Lactose starts transcription, but lactose starts transcription because it binds to the repressor. So this is hard. You may want to watch this part of the video again. Okay, this is another operon in bacteria, the tryptophan operon. Same thing. Operons control transcription, but this one is just opposite. Instead of, so just take a second and look at this one. RNA polymerase goes down RNA. Right here, RNA polymerase goes down DNA, RNA polymerase goes down DNA, makes RNA. And that's because the repressor is not attached. The repressor is not attached. Okay, well, the only thing that's opposite here, in the other picture, lactose stopped it. Well, this time, tryptophan activates the repressor. So when tryptophan binds to the repressor, then the repressor binds to the DNA. So tryptophan binds to the repressor, and the repressor stops. So in both cases, the repressor stops. The only difference in this picture and the last picture is tryptophan activates the repressor. Lactose inactivates the repressor. On both pictures, when the repressor is bound to the DNA, RNA is not made. It's a repressor, that makes sense. The repressor stops RNA from being made. Okay, so remember, operons are how bacteria control gene expression. Okay, math, Hardy-Weinberg. Remember, Q squared is homozygous recessive, and I've said it over and over. If you'll just remember that homozygous recessive is the percent of the population showing the recessive trait. Every time you see Hardy-Weinberg, the first thing you need to do is figure out what is Q squared. How do I get Q squared? It may be given. You may have to divide the number of the population by the total. But remember that Q squared is the percent of the population that's the recessive trait. Okay, P squared, percent of the population that's homozygous dominant. 2PQ, percent of the population that's heterozygous. But these two together are the percent of the population that show the dominant trait. Right? Because someone who's big A, little a shows the dominant trait. Someone who's big A shows the dominant trait. Okay, and then P is the dominant allele. Q is the recessive allele. So those are just one letter. P is like a capital letter. Q is like a recessive letter. Okay, so we're going to work this problem. In a population of frogs, green is dominant to brown. So pay attention to what they tell you is dominant. Green is dominant. Green is dominant. 25% of the population is green. What percent of the population is P squared? Okay, so what you're going to do, first thing, solve for Q squared. Always. 0.25 is green. So that means that 0.75 is Q squared. If 25% of the population shows the dominant trait, then 75% of the population shows the recessive trait. Okay, so then, so always solve for Q squared first. Then you need to solve for Q. Okay, so I'm going to have to use my calculator here. Take the square root of 0.75. So 0.75 square root. Q equals 0.86602. You're trying to get college credit. Carry it out 4 decimal places or 5 decimal places. Okay, P is going to be 1 minus that. And I got 0.13395. That's P. So all you have to do now, so let me make sure you know how I got that. I took the square root of 0.75, and that was Q. Come on. If Q times Q is 0.75, then the square root of 0.75 is Q. And the formulas tell you that P plus Q equals 1. I erased all that data. And oh, there we go. Okay, so P is 0.13395. Okay, so because P plus Q equals 1. So to find P squared, all you have to do is 0.13395 squared. And I got P squared to be 0.01795. Okay, look at the question. They're going to tell you either how far to round it, don't round to the very end, either how far to round it, or to give it in fraction, or to give it in percent. So let's say this one said to give it in a percent. Then you would put 2%. Okay, let's say it said leave it in a decimal between 0 and 1 to the 100th position. Okay, be careful on the math. Round correctly. All you have to do is read the question and do what the question says. It may say round to the 10th. It may say round to the 100th. It may say whole number. It may say fraction. Do what the question tells you to do. Just don't round till the end. Okay, chi-square. I think chi-square is important. Chi-square, you got the sum is equal to, you got to do, the data subtracted squared divided by expected. Okay, so this one was from your mock exam. Perform a chi-square test on the data for 10 minutes. Okay, so they put that 1 minute data to throw you off. Pay attention. You just want to look at the 10-minute data. Time, point in the banana experiment. Okay, specify the null hypothesis. Okay, so the null hypothesis, remember, is if there were no variables, what would you have expected? If there were no variables, what would you have expected? So you're going to expect, null hypothesis is going to be evenly distributed. Okay, so I didn't put the table on here, but pause the video, try this one for a second, and then watch me work it. Okay, so your observed data is 45, 3, and 12. Your expected data, the null hypothesis, is going to be 20, 20, and 20. Add that all together. Okay, so you're going to do 45 minus 20 squared divided by 20 plus, and you have a little 4-function calculator, so you're going to need to do this one at a time. 3 minus 20 squared divided by 20 plus 12 minus 20 squared divided by 20. Make sure you subtract, then square, then divide. Write that number down. Do it again. Write that number down. Do it again. Write that number down. Add it all together. And when I added it all together, I got, for this one, I got 31.25. For the second one, I got 14.45. And for this one, I got 3.2. So when I added all those together, I got 48.9. Okay, now what do I do with that number? That's your chi-square value. But in case this is a writing, you're going to have to use it. Okay, so 48.9 is your chi-square value. Your degrees of freedom is always one less than the number of chi-squares you had. So you had 1, 2, 3 chi-squares. So your degrees of freedom is 2. Degrees of freedom is 2. So you're going to look at your formula sheet, look at the degrees of freedom for 2. Okay, and when I look at it, I see that in the 0.05 column, the degrees of freedom for 2, the critical value is 2.99. Okay, so your critical value is, okay, so degrees of freedom is 2. Look at 0.05 column, and I see that your critical value is 2.99. That's the table value, 2.99. Everybody see? Do this, like, make sure you're looking at your table with me so that you know how to do this. Degrees of freedom is 2. That's one less than the the formulas you have. You have 1, 2, 3, so it's 2. Always use the 0.05 column. And I got 2.99. Okay, if your chi-square, which is this number, is bigger than your critical value, you're going to reject the null hypothesis. What that means is you're off 48.9. And if you're off by less than 2.99, you're close enough to be okay. So we're going to reject the hypothesis. What that means, this is easy. In this case, you're rejecting, you knew this in part 1. I'm sure you all wrote it. You're rejecting the possibility that they were going to be evenly distributed. They're not going to be 20, 20, 20, 20. They're not going to be 20, 20, and 20. Why? Because the fruit flies are going to be on the banana. They're going to be at the 45. They're going to be over there. Fruit flies are attracted to the banana. So you have rejected the null hypothesis that they were going to be evenly distributed, because they are not going to be evenly distributed. Okay, last slide, last one, and we're done. Okay, rate. If you see rate on the exam, which I expect you will, if you see rate on the exam, I don't want you to look for a formula. I want you to see the word slope. When you see rate, see slope. See 7th grade math, y2 minus y1 divided by x2 minus x1, slope. The only way you're going to miss this question is if you're not careful and do it carefully, because you know how to do this. So let's say the question asks for the slope of the line between 0.6 and 0.8. All right, I'm going to make that a little bit easier for me now that I look at those points. Okay, and as for the slope, we're going to pretend like the question says the slope of the line between 0.2, which is exactly 10, and 4.45. Or no, 0.5, it'd be 0.5, which is negative 10. Okay, so you're going to do rise over run. Okay, so on this one I would do y2, which is negative 10, minus positive 10, divided by 0.2 minus 0.5. So we're really going to do 0.5 minus 0.2. So you got negative 20 divided by 0.3. Put that in your calculator. Make sure you don't forget the negative sign on this one. You should have known it was going to be a negative slope because it goes down. Right, the line goes down. Okay, so make sure that when you see rate, you know slope. Again, round correctly. Whatever the question says to do, 10th place, 100th place, whole number, whatever, round correctly. And I put this lab data on here just because I wanted to remind you, if you're asked, this was that very first lab we did, except we used a potato, and we put a potato in distilled water in a sucrose solution of 0.2, 0.4, 0.6, 0.8, and 1. And remember, at the distilled water, the potato gained mass. So you weighed it the next day, and it had gained weight. At 0.2, the potato had also gained weight. But at 0.4, if here's 0, at 0.4, the potato had lost weight. 0.6, it had lost weight. And 8.8, and then 1.0, it had lost weight. So just remember that water moves towards the solute. Water is going to go towards the solute. So the lab, we were trying to determine the sucrose concentration in the potato. And that would be where the potato didn't gain or lose any weight. Right, so right at 0, I think looking at this picture, it's going to be like 3.5 or 0.35. That would be the where the molarity of the potato is, because it didn't gain or lose any weight. All right. Good luck, guys. I just want you to know that you're prepared to take this exam. You've studied all year. Take a deep breath. Don't be nervous the day before, the morning of. take a deep breath and do the very best you can. Good luck.